Good morning. It's March 12th, and this is your daily brief in science. Here's everything you need to know. A recent study published in the journal Blood has unveiled intriguing findings regarding frequent blood donors and their genetic mutations. Researchers discovered that these donors are more likely to have mutations in the DNMT3A gene, a gene linked to blood cell production and overall blood health. As individuals age, mutations can accumulate in bone marrow stem cells, which may lead to blood cancers such as leukemia. The study suggests that regular blood donation could potentially enhance the fitness of blood stem cells, thereby lowering the risk of developing blood cancer. NHS Blood and Transplant has acknowledged the study's findings, but emphasized the need for further research due to the complexities introduced by the healthy donor effect in health assessments. Researchers have cautioned that it remains uncertain whether these mutations actually prevent cancer, highlighting the necessity for larger sample sizes in future studies. The research team plans to expand their study to include larger samples and female participants to gain deeper insights into the implications of these mutations. The team aims to investigate the role of these mutations in the development of leukemia and explore potential treatment or prevention strategies. Dominique Bonnet, a senior author of the study, has stressed the importance of confirming these findings through larger sample sizes to better understand their impact on blood cancer incidence. The collaborative research involved teams from the German Cancer Research Center and the German Red Cross Blood Donation Center, enhancing the study's scope. In terms of genetic mutations, both frequent and irregular donor groups exhibited a similar number of natural mutations, with frequent donors showing 217 mutations compared to 212 in irregular donors. Notably, 50% of frequent donors had a distinct type of mutation in their stem cells, compared to 30% of irregular donors, which exhibited different growth patterns in laboratory settings. Laboratory experiments indicated that blood stem cells with the DNMT3A mutation grew 50% faster than unmodified cells when exposed to erythropoietin, a hormone released after blood loss. An Australian man has made history by becoming the first patient to leave a hospital equipped with the Bivacor Total Artificial Heart, as announced by St. Vincent's Hospital in Sydney on March 12, 2025. This patient survived over 100 days with the Bivacor device before receiving a traditional heart transplant, marking a significant advancement in artificial heart technology. Prior to this case, 5 patients in the United States had received the Bivacor device during an early feasibility study, but none had been discharged with the implant. Despite the promising results, the total artificial heart is still undergoing clinical trials, and its long-term viability as a substitute for a natural heart remains uncertain. The Bivacor heart is specifically designed for patients suffering from end-stage biventricular heart failure, a condition often caused by heart attacks or diabetes. With over 23 million people globally affected by heart failure each year, and only about 6,000 receiving donor hearts, the need for alternatives like the Bivacor is increasingly critical. Developed as part of the Monash University-led Artificial Heart Frontiers program, the Bivacor heart has received a $50 million government grant to further develop heart failure technologies. Utilizing magnetic levitation technology, the device mimics the natural blood flow of a healthy heart and features only one moving part, significantly reducing wear and tear. According to his doctor, the patient managed daily activities, such as shopping, while living with the artificial heart before receiving the transplant. The procedure, performed in November 2024 by cardiothoracic surgeon Dr. Paul Yance, was deemed a clinical success, allowing the patient to survive long enough to receive a heart transplant in March 2025. This achievement marks a significant milestone in Australian medicine, as the patient is the first to receive the TAH outside of the United States, where further clinical trials are ongoing. The blue-lined octopus, a small yet highly venomous cephalopod, has captured researchers' attention with its unique mating strategy. Found in shallow reefs and tide pools, this creature is recognized by its striking electric blue lines that emerge when threatened, serving as a warning to predators. A recent study reveals a startling behavior during mating. Male blue-lined octopuses inject females with venom to prevent being eaten. Males must carefully regulate the amount of venom they inject, as while females are resistant to tetrodotoxin, the potent neurotoxin they use for hunting and defense, they can still be affected. The study found that males have significantly larger venom glands than females, suggesting this is an adaptation for their reproductive strategy. Tetrodotoxin is produced by symbiotic bacteria and is stored in the male's salivary glands, making these octopuses among the most dangerous marine animals. During mating, which lasts between 40 and 75 minutes, the male's respiration rate increases, enabling him to stay alert while the female is incapacitated by the toxin. The effects of tetrodotoxin cause females to stop breathing after about 8 minutes, but they recover afterward, despite having open wounds from the male's bites. Females may lay eggs within 3 to 29 days after mating. This behavior underscores a significant aspect of cephalopod reproductive strategies, where the risk of predation by females is a critical concern. Sexual cannibalism is common among cephalopods, with females often consuming their mates, especially during the egg-brooding phase when they do not feed for about 6 weeks. The research, published in the journal Current Biology, provides valuable insights into this unique mating behavior and its implications for understanding cephalopod reproduction. Barnard's star, located just 6 light-years away, has long intrigued astronomers due to its high proper motion. First identified by E. Barnard in 1916, this M-dwarf star has been the focus of exoplanet searches since the 1960s. Recently, astronomers have confirmed the existence of 4 tiny planets orbiting Barnard's star, each with 20 to 30% of Earth's mass. These newly confirmed planets complete their orbits in just a few days, indicating they are too close to the star to be considered habitable. The research team has ruled out the presence of Earth-mass planets in the habitable zone of Barnard's star, which is crucial for understanding conditions that could support life. The confirmation of these planets was achieved through 3 years of rigorous data collection, analyzing 112 nights of observations. The team utilized the Maroon X instrument attached to the Gemini North Telescope in Hawaii to detect the gravitational effects of the planets on the star's position. These findings have been independently validated by different teams using separate instruments, enhancing the reliability of the results. The discovery of these small planets is expected to advance our understanding of exoplanet diversity and formation, particularly among rocky planets. Notably, the 4th planet is the lightest exoplanet detected by the radial velocity method, indicating the potential for future discoveries of sub-Earth-sized exoplanets. The study detailing these findings was published on March 11, 2025, in the astrophysical journal Letters, and involved collaboration among multiple institutions. This discovery builds on a previous study from November 2024, which hinted at the existence of additional planets around Barnard's star. Recent archaeological discoveries at Tin Shemet Cave in central Israel reveal that Neanderthals and early modern humans coexisted and interacted between 130,000 and 80,000 years ago. This site, active since the Middle Paleolithic era, has yielded the first complete human skeletons found in Israel since the 1960s. Excavations have uncovered some of the oldest known burial grounds, dating back approximately 100,000 years. These burial sites feature early human remains interred in a fetal position, accompanied by grave goods and animal bones. The findings suggest that the blending of Neanderthals and modern humans may have led to distinct cultural products, hunting methods, and social structures. This resulted in habitation sites that are difficult to distinguish between the two groups. Research indicates that interactions between these hominin groups were likely more cooperative than confrontational. Evidence of shared technologies and social behaviors has been uncovered, challenging previous notions of competition. Led by Professor Yossi Zeidner, the research team highlights similarities in burial practices and tool-making techniques. The presence of ochre, which is not locally sourced, points to long-distance trade or communication, indicating complex social interactions during this era. So far, 5 burials have been identified at Tin Shemet, with expectations of more discoveries as excavation continues. These findings, reported on March 11, 2025, align with earlier discoveries at Kafsa and School caves, suggesting they represent some of the oldest intentional burials in human history. Excavation efforts began in 2017 and have faced challenges due to hard sediment conditions. Nonetheless, researchers have successfully extracted significant artifacts, including a 1-ton block containing a skeleton. Zeidner emphasizes that these interactions were fundamental in driving cultural and technological innovations throughout history. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.